Good afternoon. As president of the National Math Honor Society Pi Mu Epsilon, it is a delight to introduce Dr. Trachette Jackson as our 48th J. Sutherland Frame Lecturer. It was actually Dr. Frame himself who gave the very first Frame lecture at the AMS-MAA summer meetings in 1975. For those who don't know, Dr. Frame served as president of Pi Mu Epsilon from 1957 to 1966. He was a prolific researcher and was dedicated to helping students grow professionally and achieve success in their careers. So it's very fitting that Dr. Jackson is this year's Frame Lecturer, as her own mathematical pursuits are very much aligned with Dr. Frame's. Dr. Jackson received her PhD in Applied Mathematics at the University of Washington, supported by an NSF graduate fellowship, then spent time at Duke University as a postdoctoral fellow. Since then, she has been at the University of Michigan, where she currently serves as Associate Vice President for Research, DEI Initiatives, and as Diversity and Social Transformation Professor of Mathematics. Dr. Jackson is a prolific researcher and a dedicated leader in STEM education. She's received numerous honors and awards for her work, including the following. She's an inaugural fellow of the Association for Women in Mathematics and the first African American fellow at SIAM. Her mathematical research in mathematical biology has produced many publications with co-authors that include undergraduate students, graduate students, postdocs, and her collaborators. That work has been supported by several grants, including current funding from the NSF and the NIH. She is the 2023 recipient of the Leah Edelstein Trachette Prize, awarded by the Society for Mathematical Biology for her exceptional research contributions. She's been featured and highlighted in several forms of media, including the following. The documentary film, Darwin's Extra Sense, about leaders in the field of mathematical biology, that premiered at the JMM in 2013. The 2016 book, STEM Gems, How 44 Women in Science Shine, and How You Can Too. And in the 2018 Forbes magazine article titled, Girls, If You Want to Change the World and Earn a Lot While You Do It, Try STEM. In addition to her research achievements, Dr. Jackson is also dedicated to helping students succeed in mathematics, especially women and those from underrepresented populations. Her achievements in this regard have included NSF-funded service activities to provide scholarships to support low-income undergraduate students, undergraduate research opportunities at the intersection of biology and mathematics, and a bridge to the PhD program for underrepresented minority scholars. Her success and dedication to STEM education and mentoring has been recognized both locally and nationally, including the Blackwell Tapia Award in 2010, which recognized both her research contributions and her service in supporting students from underrepresented minority groups. Pi Mu Epsilon would like to add one more honor to your list, Dr. Jackson. We would like to induct you officially into PME. If you're willing, please repeat the PME pledge after me. I solemnly promise.
I solemnly promise
that I will exert my best efforts to promote true scholarship, particularly in mathematics, and that I will support the objectives of the Pi Mu Epsilon Society. Please join me welcoming Dr. Jackson as she tells us about mobilizing mathematics for the fight against cancer.
Thank you so much. Hello everyone. Good afternoon and Happy New Year. I can't think of a better way to spend the first week of 2024 than at the joint mathematics meeting. So glad you all felt the same way. And because there's nothing I like better than getting an opportunity to speak with students, I was honored beyond words to receive this particular invitation. And I'd like to thank the organizers of this talk for selecting me and for that all too kind introduction. So I would like to spend my time today sharing with you some insights and ideas that I have for mobilizing mathematics for the fight against cancer. And what I'd like to do first is kind of orient the students in the audience to the field of mathematical oncology. Then I'll tell you a little biomedical story about the problems that I'm working on. And then finally, we'll develop a couple of mathematical models that we're using to answer some specific questions associated with molecular therapeutics for cancer. So that's the plan for today. So this was a Time magazine cover several years back, How to Cure Cancer. And the headline of the article associated with this cover was, "The hero scientist who defeats cancer will likely never exist." And they described cancer in this article as an intricate and potentially lethal collaboration of genes gone awry, of growth inhibitors gone missing, of hormones and epigenomes changing, and rogue cells breaking free. They said that cancer acts as one great armed force, attacking by the equivalent of air, land, and sea in stealth. And that last question, what are we going to do about it? What can one person do about the 300 plus diseases that we call cancer, right? So there may not be one hero. Instead, there may be many. And this article called for all sorts of expertise to come together and complement each other to fight cancer, right? And so they mentioned a lot of different kinds of expertise that would be necessary. Of course, biological scientists and medical oncologists were mentioned. A variety of types of engineers were mentioned, including biomedical engineers. And then informaticists and people that work at the genetic and genomic scale, of course, were mentioned. But a particular kind of expertise was left out. Mathematical expertise was left out. And I took that to heart, right? Because one of the things I'd like you to get out of this talk before you leave today is that people with our skill sets who are interested in mathematics deserve a seat at the table, and we can be key contributors to the fight against cancer. So you'll see that I drew myself in there because I believe that we can have something to say about this and something to contribute to this team-based, cross-disciplinary approach to cancer research that really is going to upend tradition and deliver results faster. Okay, so I said I wanted to kind of orient the students in the audience to the fields of mathematical oncology, relatively new term, which just really means bringing, computational and theoretical tools to bear against the fight against cancer. And it may surprise you to learn that researchers have been using various forms of mathematical approaches to gain insight into how tumors grow and to predict to how better treat them for over a century. So when you think about it that way, mathematical oncology can be thought of as an old discipline. But it's also at the same time new because we're reimagining and re-envisioning ways that math can be useful. So today, mathematics and theoretical approaches have been applied to every aspect of tumor growth, from the mutations that acquire in your normal cells to kick off the process, all the way down to, the transport of cancer cells in your bloodstream, and all the way to, predicting how they will respond to these intricate new kinds of therapies that are becoming available on the market. So in that sense, we have a new discipline that we're terming mathematical oncology. At first, it was thought of as a subdiscipline of applied mathematics. Applied mathematicians would use their tools to, develop models and say things about cancer. But now, the thinking has begun to migrate towards mathematical oncology actually being a subdiscipline of oncology itself. And that shift in mindset, that shift in thinking is critical, right? Now, the whole world is beginning to understand what a powerful asset mathematicians and theoretical computational scientists can be in the fight against cancer. So I want to give you a few examples as to why we need this discipline called mathematical oncology. I'm going to give you a few examples. So the first reason is that more data leads to the need for more mathematics, right? So in today's age, we are generating tremendous amounts of data. This example on this slide is data about the inner workings of cells and how cells make decisions. Because we know when cells make the wrong decisions, that's when we can get things like cancer, right? So cells synthesize all kinds of information from their surroundings, put them through these complex intracellular signaling networks with numerous feed-forward and feedback loops, that are difficult to understand and make predictions about. So if we're able to develop mathematical models of these signaling networks, right, we're able to help to integrate all the experimental knowledge and experimental data that we're gaining into a coherent picture and use those models to test hypotheses about the mechanisms that underlie these cell decision-making processes. So we may be able to predict if the cell is getting all this information from the outside world and putting it through these signaling cascades, is the cell going to decide to divide or to move or to die? And a mathematical model can help make those predictions. So that's one example of more data leading to more mathematics. Here's another more cancer-specific example. This slide is about the Cancer Genome Atlas called the TCGA. So the TCGA produced over 2.5 petabytes of data and information. And just to put that into context, 1 petabyte would be over 200,000 CDs of information, right? And this data comes from 33 different tumor types, including several rare cancers, from 11,000 different patients, their tumor tissue and their normal tissues, across 7 different kinds of data platforms. So there is a real critical need for time and cost-efficient strategies to analyze and interpret all of this data, right? We need to be able to extract meaningful information in a systematic way. It's not good enough just to have this information. We need to be able to use it to advance human health. Specifically, that means we need mathematical approaches to generate reasonable, testable hypotheses from all this data that's being generated. So we may decide to use something like more classical approaches like principal component analysis, which is just a method for exploring and making sense of these kinds of data sets with large numbers of variables by reducing their dimension to a few principal components that explain the main patterns of information in that data. So that's kind of a classical approach, but other approaches and using other kinds of mathematics are emerging. So topological data analysis methods are rapidly developing into its own subfield for the study of this kind of genomic data, right? So this is employing tools of algebraic topology to kind of understand the information contained in these vast data sets. So more data has definitely led to the need for more mathematics in the field of oncology. More mathematics has led us to start thinking about new ways of using mathematics. So we just talked about these large data sets that are being generated. Well, they can be used to guide mechanistic, multi-scale mathematical models where the system is described at different levels of complexity and resolution, and those are all integrated mathematically or computationally to answer biological questions. The way we can do that is to do that more accurately is to combine those kinds of models with all of this data that's being generated, and a new use of that is to generate virtual populations or virtual cohorts of patients to test new strategies on. So we can create these computational populations that have features of a non-uniform human population and test a bunch of strategies on them. So that's a new way of using mathematics, virtual population, virtual cohort analysis. So new ways of using mathematics is of course going to lead us to needing more computational power. Luckily for us, advances in computing power is just escalating, beyond words, right? And so we are able now to do calculations and simulations that just weren't possible a couple of decades ago when I first started this process. So what this graph on the screen is showing is a prediction of the evolution of computing power per cost, right? And in 2001, a very famous computer scientist predicted that by 2020, a few years ago, a $1,000 personal computer would have the raw processing power of a human brain. And by 2030, it would take a village of human brains to match a $1,000 personal computer. Well, I think we all know that we're not quite there yet. We don't have that power at our fingertips on our laptops. But hot off the presses, a new supercomputer is supposedly scheduled to go online in just a couple of months that will rival the estimated rate of operations of the human brain. So we're getting there, we're getting close. And this is so exciting because all of this computing power is going to lead to endless new possibilities. So more math, or more data, excuse me, leads to the need for more math. More math leads to new ways of using math. And then this new math leads us to need additional computational tools. And these new computational power is leading to all of these possibilities. What you're seeing here is an example of a supercomputer called Aurora, that scientists are using to help visualize the spread of cancer in an anatomical section of the brain before treatment and after treatment. So we can do these large-scale simulations of what's actually going on in a particular person's brain anatomy, things that we just couldn't do before, right? And here's another example. So we talked about that cancer cells can enter your bloodstream and spread. Well, there are over 60,000 miles of blood vessels in our bodies, right? And once a cancer cell enters that, it has to navigate that tortuous vascular network. So researchers are also using supercomputers to build mathematical models that simulate blood flow through these complex networks so that they can predict the path of cancer cells. So we may be able to know better where they may end up and where they may try to form new tumors in the body. So again, the need for mathematical oncology through all these examples, to me, is really, really clear. We have a lot to offer by bringing these approaches to bear. Another reason is simply that cancer is a dynamical system, right? It cuts across multiple biological scales, and that's what mathematics is made for, the study of dynamical systems, right? So it's a natural tool, a natural choice for uncovering how multi-scale processes are integrated to give us the things that we observe. So on this slide, if we look at cancer, we could think about it from all these different scales, right? From single molecules on up to the signaling cascades that may go wrong, that lead to individual cells making the wrong decisions, which lead to populations of cells doing the wrong things, that could affect an entire organ and eventually an entire organism. So traditionally, mathematics has been called on to answer questions associated with each one of those biological scales, right? And the type of mathematics used depends on which scale you're working on and what question you're trying to ask. What's happening more and more today is that we're trying to answer questions that cut across several of these scales. So the boundaries of the types of mathematics that you use are a little bit blurred because you may not need just one type of mathematics. You might have to pull in other kinds of mathematics in order to answer questions that cut across these scales. So in my work, I typically try to answer questions that cut across these three scales, from intracellular signaling networks to single cells making decisions on up to the population of cancer cells that are growing in our bodies. With an eye, in my work, always towards molecular therapeutics. I'm always interested in how can we use mathematics to help treat cancer better. So I want to switch gears and start telling you a little bit about the kind of work that I do. As a mathematical oncologist, my work is very medically oriented and biologically oriented. So in order to have you understand what the models I'm going to show you represent and what questions they are really trying to answer and the importance of those questions, I'm going to ask that you bear with me as I tell you a little story about the biology behind these models. And then I promise we'll get to some math, okay? All right. So I'm going to tell you about some of the current work my team is doing developing these multi-scale models, and we'll talk about what those mean, for understanding tumor growth dynamics, how tumors grow in their microenvironment, and for improving therapeutic outcomes when we're dealing with more than one therapeutic option, so combination therapies. That's going to be the goal. So more specifically, What we're trying to do is combine two different kinds of mathematical modeling approaches. We're going to talk about some multi-scale ordinary differential equation models, and then some agent-based models, and we'll talk about what those are also. We're going to use both of those mathematical approaches. We're going to combine that with experimental technologies that can quantify cellular dynamics and cellular interactions. So these are technologies that can actually measure single cell interactions and give us parameters that we need for the models that we're developing. And the reason we're doing this is twofold. We want to do a couple things. First, we want to develop predictive methods for improving the effectiveness of a particular kind of therapy, immunotherapy. We'll talk a little bit more about that, along with novel therapies that target very specifically mutations that drive cancer progression. And the second thing that we'd like to do is gain a deeper, kind of more robust understanding about how these mutations can impact the immune system, because that means they can ultimately impact how well the therapies are working. So that's the goal for the next little bit of time. So the models that I'm developing are general and flexible enough to be applied to just about any type of solid tumor and a wide variety of combinations of therapies. But I'm going today to take bladder cancer as the platform that we'll talk about. And I'm doing that because my collaborators are medical oncologists at University of Chicago who focus on this particular type of cancer. It's one of the 10 most common cancers in the US, and it's also one that we've just made very little progress on in the last 3 decades. So the 5-year survival for advanced bladder cancer is low. It's below 35%. Chemotherapy is the absolute cornerstone, the best we got, the best we can do. And it's not working in these advanced cases. So the question becomes, what do you do? What do you do when the hallmark of medical treatment for this particular kind of cancer, which is chemotherapy, what do you do when it's failing? It's not working. There can be lots of different answers, but one answer might be to try a targeted cancer therapeutic. Now, what are targeted therapeutics? They are just drugs that instead of blanketly killing all cells that are dividing, they're designed to interfere with and inhibit either specific cells or enzymes or proteins or molecules that experimental research has shown are necessary for the growth and progression of a particular type of tumor. So the goal of these targeted therapies is to fight cancer with a lot more precision and fewer side effects, because we're not blanketly killing rapidly dividing cells. So that's one option. Is it an option for the bladder cancers we're interested in? And if so, what could those particular targets be? Well, there are some targets for bladder cancer that we could consider. So genomic analysis has identified frequent alterations and mutations in a family of proteins in bladder cancer. And these mutations give these bladder cancer cells a fitness advantage that includes increased proliferation and increased survival. So normal bladder cancer cells are going to grow. But these mutated bladder cancer cells are going to grow faster, and the cells are going to stay around and live longer. So if we could target that mutation that gives us additional fitness advantage, maybe that would help improve where we are today with therapeutic options. So in fact, there have been some clinical trials using drugs that can stop these mutated proteins, and they're showing promise with some patients. And 2 years ago, the FDA actually approved the first targeted drug of this type for bladder cancer. So what this picture is showing, these are the proteins or receptors that are mutated and causing the cell to divide and live longer when it shouldn't. This is the drug that comes in and stops that intracellular signaling, leading to different decisions, better decisions for the cell. Okay? So that's one option. Chemotherapy fails, right? So targeted therapy could be an answer. Another answer could be immunotherapy. So this is a therapy that just arms our own body's immune system to be much, much better at its job of eliminating cancer cells, right? And this has now reemerged as an exciting domain to explore in a variety of cancers, including bladder cancer. Just to show you, it's kind of, immunotherapies have been dominating all kinds of media, and they're always in the news, from the top scientific journals, Science, Nature, even Time Magazine, to all the news outlets, New York Times, and you see things like new weapon, game changer, breakthrough, potential cure. So there's lots of excitement about immunotherapies as an option to do better, especially in cancers that we've made little progress on. In bladder cancer, these bladder cancer cells can actually suppress the immune system's response and evade their killing mechanisms. So if we give an immunotherapy, it sounds like it would be a great idea to give a drug that can inhibit the way that these cells suppress the immune system. So they have some antibodies that can do this. And they've tried it in bladder cancer. It's favorable again in some patients, but still not working well across the board. So I've got a couple more biological slides, and I promise we're gonna get to some modeling. Just to tell you that the story is more complicated, because mutations that give these cancer cells a fitness advantage, proliferation, and survival are also somehow negatively impacting the immune system, right? So what this is showing is that if you have bladder cancer cells without the mutation, you let them grow, of course they grow. If you give the immunotherapy, we get a nice amount of tumor reduction. But if you have these bladder cancer cells with the mutation and you give the immune therapy, it's like you did nothing at all. No effect. So this is beginning to hopefully you're beginning to see where mathematics can come into play, right? So there's excitement about these two potential new kinds of therapies, a targeted therapy, an immunotherapy. But there are challenges with each one that are given on their own, right? So maybe a co-acting combination of a very potent immunotherapy that boosts our body's immune system and a very targeted mutation-specific inhibitor given together may be able to give us those improved outcomes that we want to see. So if this is the case, we should see that, right? Well, here's an example of one of the very first clinical trials doing something like this, a targeted therapy plus an immunotherapy at a higher dose and a lower dose. This is 23 patients' responses, and what's plotted here is the % change in tumor volume. So what do we see? For a lot of the patients, the therapy seemed to work, right? We get large reductions in the tumor. But interestingly, the lower doses seem to do better for some reason. But then we also see there's a group of patients where the tumors just grew completely, like they weren't treated at all. And in this case, it was a high dose of therapy that did absolutely nothing. So there's so many questions embedded in this biological story that I just told you, right? So what the researchers are doing, the clinical researchers are doing is attempting to combine two very different strategies for treatment, one immunotherapy, one targeted therapy. They have clinical research that shows that the order and the timing and the amount of drug is critical. And we get different outcomes with different administration strategies. So when that happens, maybe using a data-driven mathematical model can be that powerful and practical way to help increase the efficacy of these novel cancer treatments. Maybe mathematics can help say something and show something that they weren't seeing experimentally. Okay. So let's start developing some models. The first model we're going to develop is just going to look at the impact of these cancer mutations on how tumors grow and on these combination therapies, generally. And we want to use the model to try to answer 3 questions. First, under what circumstance will immunotherapy outperform the mutation-targeted therapy? How would a virtual cohort of patients with different mutation fitnesses respond to these therapies? And can different dosing strategies improve the outcomes that we've seen? All right, so for this mathematical model, here's a schematic diagram of the things that are included in the model, and then I'll show you the equations in just a minute. So we have tumor cells And because we're modeling immune therapy, we also have immune cells. On these tumor cells, we know we have these mutated receptors that cause intracellular signaling that leads to increased proliferation and increased survival. But what we're going to do is give a drug that goes into the cell and blocks all of this, that stops that fitness advantage. We also know that these tumor cells, when they come in contact with an immune cell, can kind of bind to that immune cell and say, hey, don't kill me. I'm a normal cell, don't kill me. So we are also going to give a second drug, which is an antibody that blocks this immune suppression mechanism in cancer cells. So we're going to write down equations that capture all of those key features of the dynamics. So here's a look at what the equations kind of look like, the most important equations look like, and a demonstration of why we call this a multi-scale mathematical model. So at the cellular scale, we have a population of tumor cells that grow, first term, and die, second term. We have a population of immune cells that find the tumor somehow, begin to proliferate, and then they die, right? Or they're killed by the tumor cells. So that's one scale. That's the scale of what's going on with the two cells and how they're interacting. There's a whole nother scale of those mutated proteins. And because we're giving a therapy that acts at this scale, the therapy blocks these mutated proteins, we have to write down equations for how they behave on the cell surface. So we can write down some equations for the biological behavior of those mutated receptors. And once we get that, that information from this scale, we can summarize it as a fractional occupancy and then feed it back up into our cellular scale. So this amount of mutated receptors will lead to a fitness advantage in proliferation, so increased proliferation, and a decrease in the death rate, which is the same as an increase in survival. So all that information at the molecular scale feeds on up into our model for the cellular scale. Okay. So one of the first things we need to know is which of the parameters in this model, you'll see there's lots of parameters floating around in here, which of those parameters are most significant when it comes to determining how big the tumor is going to be. And how big the tumor is going to be, we chose an endpoint of around day 25, around 3 weeks, because that's what experimentally they typically run these experiments for. And we generated a partial rank correlation coefficient between each of those parameters and the tumor size on that last day. And what we see are 4 parameters have the most influence on how big that tumor is going to be on that last day. So, and when you look at what those parameters are, it's kind of intuitive, right? So how fast the tumor cells grow and die without the mutation, sort of their baseline proliferation and death rates are critically important. And also the impact of the mutation and its fitness advantage is also critically important. So those 4 parameters associated with growth and the fitness advantage of the mutation are exactly what is determining, most significantly, how big our tumor is going to be. All right, so we know our parameters that are the most sensitive. We know they make a big difference, so we need to estimate them extremely precisely. So this is a fit of our model to that same data we showed you when there is no mutation, and we could fit our baseline parameters associated with growth and proliferation. This is the same data I showed you earlier, and our model fit to it when there is a mutation. So if this is the baseline growth, we can estimate the parameters associated with how much additional growth and survival we get with this data set. So we have a calibrated pre-treatment model for how tumors grow. We've got our significant parameters, our sensitive parameters estimated quite nicely. All right, can we validate this model? That's the next thing. Can we believe that this model is going to produce results for treatment that are exactly what we would expect experimentally? So we ran the model, this time with immunotherapy, given in a treatment schedule that they use experimentally, with no mutation. And we had one more parameter to fit here associated with the binding of the antibody, the binding of the drug. And then we validate with an entirely separate set of data. So this is the data on how the tumor cells with the mutation respond to therapy. No data fitting here. We simply ran our model and overlaid the experimental data and got a very nice fit of the model equations to the average experimental data. So in this sense, our model was able to be validated. It predicts exactly what it should when the tumor is treated with an independent data set. So now we believe that we can trust the model's output. So let's see if we can answer some of those questions that we talked about in the first place. The first one was, under what circumstances will immune therapy outperform mutation-targeted therapy? So to answer that question, we generated this heat map. Let me walk you through it. On the x-axis, we have the impact of the mutation on proliferation. On the y-axis, we have the impact of the mutation on the survival. So these are the two fitness advantages that this mutation gives us. And then the color bar represents the difference between what happens if you give immunotherapy and what happens if you give the targeted therapy on that last day, day 25. So if you're in one of the lighter green regions, we see that the difference is negative. So the targeted therapy is performing better. If you're in one of the darker blue regions, we're extremely positive, and that means that the immunotherapy is doing better. So what does this show us? What does this heat map show us? Well, say we started up here in region 3. We're definitely in a region where targeted therapy is doing better. And notice we get that region when the mutation has a very large impact on proliferation and a very large impact on survival. So it makes sense that in those cases, the targeted therapy might be the better option. And if you're in a situation where the mutation has little effect, of course, immunotherapy might be the better option. But what's interesting is, say you start in one of these light green regions, and you look at tumors with less and less of a proliferation fitness advantage, so you're kind of just moving to the left, you eventually get to a place where immunotherapy does better. So start here and just move to the left, I eventually get into a blue shaded region. However, if I do the same thing but move down, which means I'm looking at tumors with less and less of a mutation effect on survival, moving down this way, I never switch over to the other therapy being better. So what this shows us is that the mutation impact on proliferation seems to be the key determinant of which therapy is the better option. So if clinicians can measure the fitness advantage of these mutated cells, that gives them an idea of which way to go in terms of what therapy on its own, these are single therapies, monotherapies, which therapy on its own might be the best one to deliver. Okay, second question. How will a virtual cohort of patients that have different mutation-induced fitness advantages respond to therapy? So taking that one step further and generating a cohort of patients that all are a little bit different, right? What would we expect? So in order to generate this cohort, we're going to start with something called identifiability analysis. And it really just answers the question, is it possible to estimate the parameters of our model using the data that I showed you uniquely? I already showed you we fit the model. We got some parameters. They looked great, looked good to me. But what if there's another set of parameters that does pretty good also, fitting that data, right? Are those estimated parameters unique that we had? Well, we can do some analysis to kind of figure that out. And the way we did it was using a Markov chain Monte Carlo method. We did some different sampling techniques, settled on a Metropolis Hastings sampling to estimate the best fit parameter distributions. When the best fit distributions are localized, we call that parameter identifiable from the data. We can uniquely determine a value of that parameter from the data. When the best fit distributions are spread out across parameter space, we say that that parameter is not identifiable from the data. So here are some typical distributions that you might see. This is the one we want to see. Very narrow, normal distribution, small bandwidth of parameters fit that data well. Unfortunately, this is what we see most of the time. Either edge-hitting behavior, still spanning a wide range of parameter space, or this more uniform profile where lots of parameters fit the data well. So that's what we see typically. What happens with our model? So here's what happens with the model that we just talked about developing. We have two cases because we had two sets of data. We had the data without the mutation, and we see that we get one of our parameters is identifiable, and then we get this edge-hitting behavior for the other one. And same thing happens when we add the mutation. When we're trying to estimate our fitness parameters, we get one that we can uniquely determine, and the other one we get this uniform kind of distribution. So it's not identifiable. So we weren't really surprised by that, but what we want to do is, so crux of that story, not all of our sensitive parameters are uniquely identifiable from the data that we have. So what can we do? We can exploit the fact that they're not identifiable by using that posterior distribution that I just showed you that tells me all the combinations of parameters that can fit the data well, to generate a computational cohort with all kinds of different tumor properties that we can use to test different dosing strategies on. So just to go back really quickly. So we've got these distributions of parameters that all fit the data well. We can randomly sample from all of those distributions within ranges from all of those distributions. And then each parameter is going to represent a different patient, and that patient's going to have a different level of tumor growth and mutational fitness. Okay? So this is one of those new ways of using math that we talked about, generating these virtual cohorts, exploiting things like identifiability to do that. Okay, so here's an example of a simulation of a virtual cohort of patients, and we're looking to see how they would do on different therapy, with different kinds of therapy. So first of all, this is a survival plot. So this is a fraction of our population that survived for the 25 days. What we see is that if you don't treat at all, the fraction of that survive by the end is very small, only 14%. If you just give the targeted therapy, we can improve that, almost more than double it. If we just give the immunotherapy, we do much, much better. And that's because we were in a region of parameter space where the fitness level for survival greatly outweighed the fitness level for proliferation. So that meant immunotherapy should do better. But then if you give this combination therapy with this schedule, co-treatment, just give them both together, according to each of their own dosing recommended strategies, we can actually increase the fraction of the population. All of these tumors are different, remember, but the fraction of the population that actually survives with the combination therapy is getting on towards 90%. So much, much better with combination therapy. And the final question for this model is, can different dosing strategies improve combination therapy? So we just showed you an example of co-treatment, and that's what they always try first, right? Let's just try giving these two drugs together. But it also might make sense to pre-treat with one of the drugs. So this Schedule 2 is pre-treating with the immunotherapy, and the pre-treating with the targeted therapy, and the Schedule 3 is pre-treating with the immunotherapy. Let's just see if those make any more sense. So what this graph is showing is that if you don't treat, of course, the tumor grows. The baseline schedule of co-treatment, we can get a 31% reduction by 25 days. With our second schedule, that's reduced quite a bit. We only get around a 13% reduction, so this didn't work. Put a big X through that. And then for our third dosing schedule, pre-treating with the immunotherapy, we actually get up closer to 30% reduction again. And all of these results persist even in parameter regions of that heat map where the targeted therapy does better than the immune therapy. So that's interesting to know. It doesn't matter which region in parameter space you're in, these results generally persist. So why does the title of my slide say pre-treatment with immunotherapy is the better therapeutic option? Shouldn't I be comparing 31 to 27? 31 certainly seems better than a 31% reduction certainly seems better than a 27% reduction to me. But if you take a look at this treatment schedule and count up the doses, we have 15 doses of the targeted therapy and 6 doses of the immunotherapy. We did just as well with only 5 doses and 4 doses, so a lot less drug, gave us just about the same level of tumor reduction. And my medical oncologist collaborators tell us that that means that right there is enough to give this therapeutic option the bump over the baseline schedule. Using less drug, definitely less side effects. And if we would have carried this out to the full 15 and 6, we would have definitely ended up with a much higher tumor reduction than that baseline schedule. Okay? All right, so on to the next kind of modeling we were talking about. So we have just done a pretty in-depth look at how cancers grow, what possibilities for treatment there are, and which treatment's going to do better when. Now let's take another look at these mutations more specifically and their impact on the immune system, and try to answer this question. How do different assumptions about the impact of the mutation on the immune system affect the outcomes we just saw for those therapies? No one knows. They don't know what these mutations are doing to the immune system. They just know that they do something to make the immunotherapy not work. So we can test some hypotheses with a model. We're going to use a different kind of model to do this. So now we're going to develop a multi-scale, 3-dimensional, agent-based modeling framework to try to answer this question. So in an agent-based model, every single cell is modeled as an individual entity with its own set of rules and opportunities to take in information and to decide what to do. So in our case, we have our 3-dimensional space partitioned into voxels, and we start with a small number of cancer cells in the middle of that tissue and no immune cells around it. Those cells can grow, divide, move, all that kind of stuff, right? Interact with anything in the environment, and we make our updates to what's happening in discrete time steps. So for this model, we have lots of different agents or individual cell types that we're looking at. So we have 2 different types of tumor cells. We have immune cells, that's CTLs. We have immune cells that are killing in 2 different ways. And then the immune cells could also be deactivated, meaning they've been around a long time and can no longer kill, or they can be sort of unengaged, just floating around, haven't found a tumor cell partner. So these are our different agents. So when you see those different colors pop up, that's what they mean, and I'll remind you of those in a minute. So again, no one knows what the mutation's doing, so we can test some hypotheses for our collaborators. The 2 most reasonable hypotheses are that the mutation is affecting recruitment of immune cells. So when you have the mutation, fewer immune cells arrive and find the tumor. Another plausible hypothesis is that there is a killing effect, a cytotoxic effect. So when you have the immune system, when you have the mutation, the immune system is not able to kill the cancer cells more efficiently. They still come, they find the tumor. It's just when they try to engage with the cancer cell, they're not able to kill it. So we have all of these possibilities to test. No effect of the mutation on the immune system, which we know isn't happening, but we can still test it. Both of these things are happening. Maybe there is a recruitment and a cytotoxic effect, or maybe there's one or the other. So we can test all of those hypotheses. So first I want to show you a still picture of an outcome of the simulations of this agent-based model when we're using combination targeted and immunotherapy. We can, when the mutation has no effect on the immune response, this is what we would expect, right? No cancer cells left, only immune cells left. Look at the tissue, looks pretty clean in the 2D slice. Right? So if the mutation really did not have an effect on the immune system, this therapy would work wonders, right? Immunotherapy would work wonders. However, if the immune system impacts both recruitment and the killing mechanism, we always get escape. All cell types persist, the tumor keeps growing, we get a slice of tissue made up of every type of tumor cell in different spatial locations. Okay? Now I want to show you a couple of movies of what this might look like, because those stills kind of hide some features. So on your left, it says elimination, and on your right, it says escape. But look at that elimination picture. It seems like it's growing to me. just as well as the escape picture, but look what ends up happening. So these are all our tumor cells growing, immune cells killing in 2 different ways. Eventually, that tumor that was growing and doing seemingly really well was eliminated by the immune system. And eventually, the other one on your right could not, the immune system could not handle the growing tumor. So looking at those stills kind of hid the fact that that tumor was growing during that entire time and eventually became eliminated. Okay, let's see. Get off of that. Okay, in the interest of time, I'll just go through this really quickly. This just shows some of the things that this agent-based model can capture, which are kind of clinically relevant about the differences between elimination and escape. We can look at, on your left, everything that's happening with the tumor, and on the right, the tumor cell number and the immune cell number when elimination occurs. We can look at where the immune cells are and how many they are when elimination versus escape occurs. We can track everything that's going on, how many times the cells divide, die, how the immune cells are killing. We have all of this information from this computational model. So with all of this to bear, let's see if we can answer that question that we had. So looking at our hypotheses for what happens with the effect of the mutation, right? Either a recruitment effect or a killing effect. Here, we have just the null hypothesis of neither one in purple, just a killing effect in red, just a recruitment effect in this yellowish green color, and then both hypotheses active in blue. And we get very different outcomes if we look at, this is on the x-axis we have time, on the y-axis we have how big the tumor is. And then we have all of our different types of hypotheses here. Notice, we can clear the tumor, but only when we're red or purple. Only when we're red or purple. That means only when the mutation does not inhibit the recruitment effect can we actually clear the tumor. The yellow and blue cases, in both of those cases, the immune system was inhibiting the recruitment effect. And in those cases, we got the tumor escaping. Right? So we can take one further look to see what's happening with the immune cells under those same circumstances to see if there's something about what the immune system is doing that could be leading to those kinds of results. So again, purple and red are the situations where we cleared the tumor, and we notice a steeper rise early on of the immune onslaught, right? So the immune system seems to come online a little bit faster and eventually clears the tumor. It takes a while. The tumor does grow and get kind of big, but then it eventually is cleared. And then for the yellow and the blue, we see a slower rise in the immune system, but eventually the immune cells get there. We've got lots of immune cells around, but that's still not enough, right? They are not able to change the fate of that tumor. So elimination occurs due to, it seems like in the elimination cases due to that early onslaught, that early increase in the immune cells, but escape happens even despite the fact that we end up with a large population of immune cells at the end. Okay? So we've got this new information gained from this model about what impact the mutation has on the tumor and what its importance is that we share with our experimental collaborators. The last question, again, is about those dosing strategies, and then we'll come to a close. Remember, we had co-treatment, we had pre-treatment with the immune therapy, we had pre-treatment with the targeted therapy, and just to round things out, we looked at it, what if we just shift and give both treatments together a little bit later? So we're calling these each different schemes. So we have co-treatment, we have pre-treatment with the immune therapy, we have pre-treatment with the targeted therapy, and then we just have that shift as version 4. So now we want to see, does the treatment outcome depend on which hypothesis we choose for the mutation's effect on the immune system? Okay? So if we give all of these dosing strategies to each of these situations, this is no effect of the mutation, this is both effects, this is just the killing effect, and this is just the recruitment effect. It turns out if you want to get the smallest tumor size by the end of your treatment schedule, scheme 2, across the board. It does not matter which hypothesis you choose, pre-treatment with the immunotherapy is the best choice. So it really doesn't matter what the tumor cell is, what the mutation is doing. The model is predicting that the dosing schedule should always be pre-treatment to get the best response. Okay, so let me wrap up and leave some time for questions. We're doing a lot with this kind of platform, making significant progress. What we're doing now is investigating tumor heterogeneity. You saw that in the model, different kinds of tumor cells. You already saw different immune cell killing mechanisms has already been incorporated. The next thing we're going to look at is variable mutation expression. So what if the mutation is weaker or stronger, and what impact that may have on cells. You saw us test a couple different traditional types of dosing strategies that oncologists use: co-treatment, pre-treatment, post-treatment, but there's a whole lot of other ways you can give these therapies. So we're really working now to predict an optimal strategy, which may be very different from those that I showed today, using some machine learning techniques. And the good thing about the models that I showed you today, both the ordinary differential equation model and the agent-based model, is that they can easily be adapted to study any therapy that targets immune checkpoints like the one we talked about today. And you can also easily adapt this to another type of cancer: breast cancer, lung cancer, all that have mutations that are known to be driving forces. So it's a very reliable and flexible modeling approach. And it does actually have some translational value. So it moves beyond just basic science information that we're gaining. The experimental and computational studies combined that we're doing are able to test these clinically relevant hypotheses about how best to give and optimize these kinds of interesting and brand new combinations of therapy. So with that, I'll thank you for your time and your attention, and see if you have any questions.
Thank you so much, Dr. Jackson. We do have microphones set up here in the aisles, and so if you have questions, please come forward to a microphone and she can answer those.
Hi, I wanted first to thank you for a really great talk.
Thank you.
So my question is regarding the case of elimination. It was very interesting that the tumor is also growing. So I'm kind of wondering what is happening in that case. Is it that the cancer cells, even though there are more of them, they are becoming more susceptible to being killed by the immune cells, and so eventually they're becoming weaker at the point where the immune cells are able to wipe them out?
Yes, thank you for that question. So for that particular simulation where we showed the elimination after the tumor had grown really large, that was for a case where the tumor was made up of just what we called in the slides high antigen cells, which are very good at calling the immune system. So these cells call the immune system much better. And I don't know if you were able to notice this in that simulation, but there were many more immune cells online. So even though the tumor had begun to grow and grow and grow, it was also calling and calling and calling the immune system as it got bigger and bigger and bigger. So eventually the immune system was able to get enough of a ground hold on the cancer to eliminate it in that case. The second simulation that led to escape was a situation where the tumor cells were not so good at calling the immune system. So they were more evasive of the immune system. So as that tumor got bigger, it wasn't calling the immune system to the location as much. And so the immune system never was able to get a foothold strong enough to defeat the cancer.
Yeah, so question here.
Oh, sorry.
So very nice to show the pre-treatment is always better for immunotherapy. How about the tapering? If we want to remove, did you see the modeling, which one during the tapering, remove, stop one drug versus the other? Which one would be more beneficial?
Could you repeat that? Are you asking if we only gave one drug, which one would
You after the treatment, you want to stop the treatment. Do we stop one
That's
versus
a good
the other?
question. That's a great question. That is a great question. We have not simulated a stop treatment scenario to see if we could, at some point, maybe just stop the targeted therapy or just stop the immune therapy to see which one would do better. But I think I'm going to have some students run that simulation now to see what happens. Thank you.
Thanks.
Yeah.
Thank you for an amazing talk. I just want to ask about how you decide what mutations to look at in like predicting treatment outcome. Like, do you go by gene or like specific variant within the genome? And like, how do you decide? Do you go through like a list of different genes, or is it more like functionality, like clinically driven?
Thank you. That's a great question. So I have an excellent team of medical oncologists at the University of Chicago who do that part. But what they are looking at are genomic alterations and receptors on cell surfaces. So it's functionality. So the function of those receptors has changed. And that's what's allowing these, signaling cascades to lead to this fitness advantage. So yeah, they look at the functional genomic level.
Hi, thanks for a great talk. I have a question because your model focuses on T cells, which are part of the adaptive immune system. If you had thought about exploring cells that are in the innate immune system and adaptive immune system and kind of using those in combination therapy, and maybe the ordering of those treatments could give you a different outcome.
That is a great question, and yes, we have a separate group of work that looks at both the innate and adaptive immune response for brain cancers, and you are absolutely right. The innate response is critical for calling the adaptive response, so you do get slightly different results if you, especially for brain tumors, if you explicitly model both. In this model, the innate immune system is implicitly modeled in how we, it's in a lumped parameter in this model. It's not explicitly modeled, but we certainly could do something very similar here.
Hi, it was an amazing talk.
Thank you.
My name is Yashleen, I'm from SF State, and my first question is, just out of curiosity, was the TCGA thing was taken from the DNA components, the ATCG? I just made the connection and I was just wondering.
That's a good question. I'm not sure. Yeah.
Yeah, because those are the exact same components for the DNA, and I
Yeah.
was like, wow, that's interesting.
Yeah, I agree. Yeah.
My second question is for the cellular scale models, when you use the differential equations, were you working with the SIR model or SEIR model?
Oh, that actually is a good question. So these are neither SIR or SEIR. So those are really classical models in infectious diseases, and sometimes people have actually tried to model tumor growth in that way, but the models that I showed you took kind of a first principles mechanistic approach to what cancer does, which is a little bit different than the SIR, SEIR approach. So they weren't either one of those, honestly.
Oh, okay, wow.
Okay.
My last question is, I'm reading your paper for the new computational approach for the ABM models, and I was just wondering if you use the global method here for the ABM or the local method.
Thank you for asking about our global method. I'm always excited when we develop new computational approaches, and the global method is one of them. And we definitely used it throughout those ABM simulations. It sped things up for us quite a bit.
Okay, perfect. Thank you.
Yeah. Oh, sorry. Oh gosh.
Thank you so much for the really amazing talk. And I was just curious, as a sort of early career researcher, I've been trying to read more about different biological systems, and cancer in particular seems really daunting as to amount of stuff there is to read.
Yes.
And there is also the collaborators will help you figure it out, but to be able to get collaborators as an early career researcher, so it feels like a chicken and egg problem.
Yeah.
So I wanted to check if you had like any suggestions or how you approached that problem when you were starting out.
Yeah, that's a great question. So not to date myself too much, but when I started out, I think potential collaborators were very skeptical about what can math do for me, I think you're in a different world today, which is great. So if you're open to just talking to experimentalists and people who are doing kind of the basic biology work in cancer, what I found that today, they are very receptive to telling you about the problems they work on, and then it's up to us to kind of see the mathematics in it, because they won't necessarily see it immediately. So we have to, be able to put ourselves out there, speak to people across disciplines, and then see the mathematics in what they're doing, or see the mathematical problem or challenge, and then articulate that back to them as, I think a computational or mathematical or theoretical approach could help you answer that question in this way. So it's really about first just starting a conversation and listening to the problems that cancer researchers are facing, and then kind of hearing how you think your skill sets could be of help.
That's very helpful. Thank you.
Yeah, thanks.
All right, and one more final question.
Thank you for your talk. I wanted to ask you, in what way is your work specific to solid cancers and does not apply to blood cancers? Because it seems like the pathways and then the way immunotherapy and targeted therapies work, they work there as well, and the measurements are more precise in blood cancers.
That's a great question. So the reason these are for solid tumors is that we have them growing in a geometry of a tissue instead of in the blood. That's the only real reason. So the measurements, the signaling cascades, even some of the interactions with the immune system can all be the same across blood-borne and solid tumors, but the geometrical constraints of it growing in a tissue is kind of what makes this more appropriate for a solid tumor. We could certainly change that structure really easily to make it applicable to like a blood-borne cancer like a leukemia or something like that.
Thank you.
Dr. Jackson will be in room 204 nearby to answer more questions in person if you want to, but otherwise, let's give her a final round of applause.